hi to everyone that's joined. We're just going to give it a couple more minutes and then we'll get started. Okay, I guess we'll get started. Thanks to everyone for joining today, whether and listen to the presentation if you're listening to it recorded later on. I'm Dr. Stephen Richardson. I'm a senior lecturer based in the Faculty of Biology, Medicine and Health. And I'd like to introduce Professor Julie Gough, who is chair in the Faculty of Science and Engineering. So the purpose of today really is just to talk to you about, let me just share my screen. Advanced materials in medicine, and some of you may be aware of AMM already. Many of you may not, or may not be totally aware of what it is that we do. So we're going to run through that. We're going to give you some information about the activities that we run, who we work with, and then I'll hand over to Julie, who's going to give some examples of the sorts of work that are going on across AMM. We're very broad in the things that we do within AMM. The examples we give today are not exclusive. They're not the only things we're doing by any stretch of the imagination. They're really just to give you a flavour of the sorts of activities that are going on. And what we really wanted to get across was the importance that if you've got an idea or you have a clinical unmet need or an area where you think there might be a solution that falls under the broad remit of AMM, activities that you get in touch with us and we can try and help point you in the right direction. So if you have questions today, we might not be able to answer all of them, but we can certainly go away and find answers out. We can certainly try and help you moving forward. On the last of Julie's slides is our contact details. For AMM, our email addresses personally are on this slide. We'll make sure all that information is circulated afterwards as well. But please do get in touch with us if there's anything of interest to you that you hear about today or you think we might be able to help you with something. So what is AMM? We are a cross-faculty activity initiative funded within the University of Manchester, and we span both the faculties of Science and Engineering and the Faculty of Biology, Medicine and Health. And as I've said already, Julie and myself are co-directors, one of us from each faculty. We have a project manager, Nicola, who we share with the Pankhurst Institute. And within AMM, we have 4 core themes of biomaterials, which is headed out of FSC by Dr. Marco Domingos, tissue engineering, headed with FBMH by Professor Sue Kimber, bioelectronics in FSC by Dr. Alex Casson, and nanomaterials led out of FBMH by Professor Kostas Kostarilos and Dr. Sandra Vranic. And as I say, we're going to give some examples of the work we do in each of those 4 themes. A lot of those themes and the work that goes on span multiple of those themes and other areas outside those a little bit as well. In terms of the network itself, we have over 100 academics and researchers across the 2 faculties that are linked either directly or indirectly to AMM, meaning that their work is directly aligned to the sort of work that AMM covers. And we don't work in isolation. We reach out across different institutions within the university and beyond. And really, we're looking for new opportunities to drive this agenda in advanced materials in Manchester. So some of you might be aware of this sort of slide, which is the translation pathway for health and care technologies, spanning all the way from very basic research through initial application and proof of concept, through then development and feasibility, and then into the healthcare setting for deployment ultimately and scale up and scale out. and different activities going on that are different TRLs, so different points in this pathway. So where does AMM sit within all this? Well, actually, we like to think we cover quite a lot, certainly this R&D pipeline, all the way from basic research through proof of concept into initial development and feasibility work. And on this schematic, we are all the way on the left over here, although as I say, I think we span a lot further across than that. We sit really with digital futures and data science and AI, and these three areas fall under the broader remit of the Pankhurst Institute, and I'll talk a bit more about that in a second. And we work really with colleagues in Translation Manchester, but we also work with BRC and have links into MAHSC and Health Innovation Manchester in order to make sure that the work that we're doing is of clinical relevance, and that where there are clinical needs, we're trying to find solutions for that within the university. And we also, I'm going to talk about now, is a work that we do with Royce as well. So this schematic just really summarises this pipeline, and it's not linear in the way that it's necessarily depicted here, and each project is a bit different in terms of who we work with. But fundamentally, we want to try and identify clinical unmet needs. So that's talking to the clinicians across MAHSC and Health Innovation Manchester to see where those needs are and see how AMM might be able to help there. In terms of the core of what AMM does, we foster and facilitate these collaborations. The work that we do is interdisciplinary, so it spans both faculties and across into the clinical and commercial environments as well. We focus really on early phase research, but trying to push work through that translational pipeline. And I've talked about those four themes already of biomaterials, nanomedicine, tissue engineering, and bioelectronics. And we'll expand a bit on each of those themes later. The Henry Royce Institute is a national institute that offers access to state-of-the-art facilities based here in Manchester, but open nationally to researchers across the UK. The idea being to enhance manufacture, characterization, and testing of materials and devices. So really trying to facilitate this push through translation ultimately into application. And we work as well with the Christie Pankhurst Institute for Health Technology and Research and Innovation. And they offer whole life cycle translational support. So all the way from development and applicability of ideas with funding, with health economics and regulation, clinical trials and commercialization, ultimately with the aim of accelerating deployment of needs-led health technologies. And finally, Translation Manchester, which offer a whole network of support facilities and funding with expertise in overcoming any translational bottlenecks and again accelerating clinical translation. So all the activities that we do across these different areas in the university really all have the same ultimate aim, which is to identify new areas of research, to drive that research and ultimately deploy those technologies back into the clinical setting to help patients and help clinicians and the healthcare setting generally. As I mentioned before, we also work with members of the BRC, and Julie in particular has been involved with MAHSC in the disease complexity research priority area. And AMM is involved in the next generation therapeutics theme of the new BRC bid that's currently in at the moment. That's around £64 million in total for that bid. And the decision should be announced sometime, I think, in May or June. But again, identifying and highlighting the fact that materials and medicine is a really important and growing area, not only for Manchester, but across the UK. And Manchester really is a leader in this area in the UK. And we want to try and promote that as much as possible. So that's a bit about who we are. What do we do directly? So we regularly run strategic workshops. Over the last couple of years, we've been doing these online primarily due to COVID, but we've had some really successful meetings around a number of different areas. The first one of these we ran was on cardiovascular, but we've run musculoskeletal meetings, meetings around inflammation and immunology and barrier function, on neurosciences, and we've got a meeting coming up soon on graphene in its application as a biomaterial in tissue engineering. And there's other graphene activities and nanomaterial activities around their translation. And we've run recently a couple of workshops looking at co-cultures and organoids. with applications, for example, in cancer, and how you get these cell-cell interactions and how we can better model that to better understand disease, but also in terms of other diseases and other areas of basic science like stem cells and their differentiation, applications in kidney and lung and gastrointestinal organs, and modeling that in vitro and what the technologies are we have in Manchester to do that at a really high level. So those meetings will go on, and those workshops will go on in the future, and hopefully soon we'll be back face to face. And if you're interested in finding out about those activities and anything else that we do, again, the last slide has our contact details on, so we can add you to our mailing list so that you can find out more about that. In terms of successes and ongoing activities in terms of funding, we are part of the EPSRC PhD training program, which is the Centre for Doctoral Training and Advanced Biomedical Materials. And Julie's going to talk a little bit more about that later as well. And colleagues within AMM are heavily involved within the International Alliance for Cancer Early Detection, and a number of the activities there really driving cross-faculty collaborations and activities and the application of models and materials which can improve early detection of cancers. AMM itself, we have a postdoc based between Julie and I's lab, and I'm going to talk a little bit about the work there, but really trying to demonstrate some of the platform technologies that we have within the Royce to model tissues and model disease, and look at then how we can use that and the facilities we have in Manchester to better understand disease processes and to test new drugs and new therapeutics. And the work we do is not just internal in Manchester. We also look externally, both nationally, but also we're heavily involved with some strategic collaborative links internationally, particularly with Melbourne and Toronto, but also with ISSC Bangalore and Tata, and the British Council India and other organisations in different countries around the world, where we can bring collaborative money in and bring skills from elsewhere to Manchester, but also offer the expertise and the equipment we have in Manchester to others to try and develop new therapeutics. So I've mentioned the Royce, we work very closely with. I wanted to just expand a little bit on some of their facilities and equipment that they have, and how they can be useful in developing new materials for medicine. So additive manufacturing and biofabrication is a major area within Royce, and they have a broad range of capabilities, either for early stage or more translational research where you want to scale up projects. The equipment and the facilities that they have there span a whole range of resolutions and speeds of manufacture, and they're really unique in the UK. So it's the hub building, which is based on the main campus, the new Royce building, houses a lot of this equipment, which is state of the art, and the only place in the UK you can find much of it. Much of that equipment is turnkey, so you can come in and use it, although a lot of it is being developed, and then there are new elements of that working with the manufacturers to try and enhance that and work out how we can improve that equipment and offer new opportunities. We have that optimized and are still optimizing it for use with a range of materials, whether that be biomaterials or biological materials. And as I say, there's a range of resolution and build rates available. And so I'm not going to go through this in much detail, just to say that we have a range of different abilities, from selective laser sintering all the way up here, down to electrospinning at this end, and some brand new state-of-the-art multi-axial robot-controlled electrospinning equipment within the Royce. And I'll talk a bit more about bioprinting in a couple of slides' time. We're talking, as I say, about nanofiber spinning and electrospinning. We have some disruptive spinning technologies within the Royce, including high throughput multi-needle electrospinning. We can create these structures within environmentally controlled conditions, and we can do roll-to-roll and continuous fiber yarns. We can create aligned fibers, and we can create hierarchical and gradient structures. Now, that might not mean a lot to some people. What it means is we can create some really complex structures that have a broad range of applications. So, and we can create them from a range of materials, including hydrogels and conductive fibers as well. So if there's an application where you think this spinning technology, this ability to create fiber yarns or sheets or rolls or tubular structures might be of use to you, again, please reach out and we can see which of the bits of kit that we have might be beneficial to you. Once we've created these materials, we also have the ability to test them mechanically. So we have multi-mode simulation and analysis equipment within Royce to do tension and compression and perfusion experiments, and do all of this under physiological conditions as well, of pH, of carbon dioxide and oxygen and pressure. So we can really test these materials, whether they be for modeling of disease or whether they be for implantation, under physiological conditions. We can also run multi-specimen fatigue testing to test up to 12 samples at once and accelerate that degradation and test the mechanical properties, but test the degradation products as well. From a more biological perspective, we can look at biomimetics and cell substrate interrogation. So looking at strain in 2D, in temperature controlled and environmentally controlled chambers. And we can do high throughput simulation and map that with imaging of constructs and cells in culture using some of the equipment that we have. So all the way through from basic science and cell work through to materials testing for implantation. We can do a whole range of tests there, and all these facilities are readily available in Manchester. And finally, on this aspect, we've got organ-on-the-chip capability. And what this allows us to do is to have multiple cell types in a closed loop system so that we can look at how these cells respond, for example, to drugs. And because we can have different cell types from different organs, different tissues, we can look to see, for example, if you have a drug that you want to work on one organ in the body, how it might affect, for example, the liver or the kidney or the heart or another vital organ, to check that there are no adverse cell responses to that drug. So that, again, this equipment's available. It's quite new in Manchester, so we're looking for applications for this at the minute. And then last slide for me really is just an example from a basic science perspective of some of the bioprinting work that we do. And so, as I've mentioned, the fact we have this ability to 3D bioprint multi-materials, and what it allows us to do is create complex structures. And I've got a couple of examples on this slide here. So on the right here, this is a meniscus. And what we can do is we can image this, and this is a 3D laser scan of this particular piece of tissue. But we can also take clinical images. So we can take MR images and CT images of human tissues and create highly accurate material-based models of that, the mimics of that, either for, as I say, tissue modeling and disease, studying diseases and drugs, or potentially for implantation as well. And there's a whole range of materials available to us for that. So in this case, where we've scanned the meniscus in, we can then print that, and we've included on the end of this some suture tabs. And then we can image this again to make sure that it represents this original architecture and size and structure that we've scanned originally. What that allows us to do then is create hierarchically ordered complex structures based on clinical images that are patient specific. And on the left here, I've given an example of how we can use that then to engineer tissue in the laboratory. So this is an example of intervertebral disc. So we have a highly aligned, ordered collagen ring structure called the annulus fibrosus. And we can model that, and we can design that using a CAD-based approach. We can then print this annular ring structure using collagen, and we can get alignment of that collagen. But what we can also do within this is load cells into these materials and pattern cells spatially in 3D through these constructs. So in the bottom here, you've got an example of one cell type in red, of the MP region of the intervertebral disc, and then that's surrounded by annulus fibrosus cells in this annular ring structure. And we can create interfaces there, and that allows us to model and study cell-cell interactions, but also engineer these tissues that are as faithful to native tissue as we can. And if we want to use that then to study development or disease, we can create tissue stiffness gradients, as in this example here, where we've gone from soft to stiff, depending on the material that we print with. And we can add in bioactive factors to these materials as well. So we can model development, we can model disease. And once we've engineered tissues, we can then use those tissues to test drugs, as well as to understand biological processes. And this difference as well can be used to control cell fate and function to engineer healthy and degenerate tissues. So Julie now is going to move on and talk about more translational projects, but I just wanted to give you an example of how we can use this from a basic biology tissue engineering perspective, and how these advanced models then can be used across a broad range of different tissues. And we've got colleagues working in barriers and immunology, in cancer, and in vasculature, and a whole range of other different areas. So if there's an application where you've got an interest in the basic cell biology or in drug testing, we have a range of ways that we can help you develop those ideas and take them forward. So I'm going to stop there and stop my share screen, and I'll pass over to Julie.
Yeah, thanks, Steve. Okay, I'll just share my slides now and get going on some further examples of some ongoing projects across our academics through AMM. So one of the projects that we've got going, which is actually extremely wide-ranging, and that's using Alberto and Aileen's Manchester Biogel peptide hydrogels, and quite a few academics across Manchester are working on this, not just me, Alberto and Aileen, for many different applications. So this is just a really brief overview of some of the properties of those peptide gels. So they're really easy to make in the lab, and we actually now get them from Manchester Biogel, just because it's easier for everybody to get them from the company. They do quality assurance and everything, and we get the same results every time. So they are self-supporting hydrogels, and if you look at them through an electron microscope, they actually really mimic the extracellular matrix of many tissues and organs. So that's why we think they're so useful for many tissue engineering and in vitro drug toxicity testing systems. So we have a huge amount of research going on across Manchester with these gels. Pretty much every tissue and organ in the body, I think, is probably covered at some point across Manchester. We also have an interesting project led by Sarah Cartmel and some clinicians in Manchester. This is looking at tendon repair, and they're setting up a spin-out company called Retendon at the moment. So they're manufacturing a novel tendon repair augmentation device made of biodegradable polymers. And this has gone through quite rigorous animal testing and degradation testing and cyclical loading testing. And they're trying to get it into a clinical trial at the moment. So that's quite an exciting project that seems to be getting quite far along our translational pathway. Another good story is the polynerve story, which was led by Adam Reed and myself and others. Our peripheral nerve guide conduit has gone through a phase 1 clinical trial in 16 patients, and we got nice favourable results. And again, this is now trying to be spun out of Manchester with the help of the Innovation Factory. So the nice thing about this is obviously similar to Sarah Cartmel's Retendon, is that it's made in Manchester and then translated through. So the idea of this particular conduit compared to other commercially available conduits is that it's got these aligned grooves on the inner lumen, which are specifically designed for the Schwann cell response, which then leads the axonal regeneration. So hopefully that will be a clinical product in the not too distant future. We've got a lot of bioelectronics work going on across AMM, and Alex Casson in the Faculty of Science and Engineering is collaborating with many people across FBMH, and he's really focusing on electrophysiological interfaces with the body. So what he can do is he can make various devices that interface with the skin and do many different types of recordings across many healthcare applications. So we've got things like non-invasive interfaces. We've got things for looking at chronic pain and sleep disorders and autism. So really flexible, wearable bioelectronic interfaces. And some of these can be actually almost tattooed onto the skin, and there are many different applications of these devices. And other colleagues, along with Alex, such as Eamon Song and Patrick Gaydecke, excuse me, are also looking at similar flexible devices and other bioelectronic devices for sensing things like cancer and looking at improving patient outcomes, basically. And also we've got Henry Lee in FSE as well, and Vincente Mercadillo also looking at sort of fabric-based sensing devices, sometimes with graphene and sometimes not. So there's a whole range of bioelectronics devices and research going across Manchester. It's quite a popular area at the moment. We've got some really interesting research coming out of Sam Jones's lab. Some of you may know of his antiviral work. So he's making cheap, easily manufacturable virucidal antivirals, and he's been testing these against a variety of different viruses, including SARS-CoV-2 with the help of Public Health England. And he's got some really interesting data coming out and is seeking clinical collaborators. So if anybody's interested in antivirals, do get in touch. Of course, we've got to talk about graphene a little bit more and other 2D materials. So we've got a lot of research going on in Manchester, of course, into graphene, and a lot in the biomedical materials area as well. So one good example coming out of Kostas Kosterolos' lab, along with colleagues such as David Coop and Rob Wicks and Thomas Kisby, are making flexible graphene devices for brain recordings and helping the surgical removal of brain tumours. So these are really useful devices, and they're going into a first-in-man study as well. Further applications of these are in a variety of things such as Parkinson's disease and epilepsy, for example. Further uses of graphene and other 2D materials, we've got Aravind in the Department of Materials. He's making sensors, really. So this is an example of a point-of-care immunosensor based on a quartz crystal microbalance with a graphene biointerface. And he's able to use these sensors to detect antibodies and markers of disease. I'm not going into these in too much detail, but he's looking at certain diseases and being able to detect them using his graphene devices. We've got a lot of projects, like Steve said, in all these areas, and these are, you know, we can't show you all of them. We've just picked out a few interesting ones. As Steve said, we've got over 100 academics across the AMM network, so please do get in touch if you have any ideas or see anything of interest, and we can put you further in touch with our colleagues. We've got many opportunities for collaborative research. If you want to contribute to some of the existing projects, or we can come up with new ideas for new projects. We're always looking for researchers and clinicians. And we also do a lot of helping identify and matching up researchers with clinicians to help get your studies off the ground and progress through to clinical translation. So we've got my slides have decided not to carry on. That's a bit strange. I'm just going to stop sharing a second and start sharing again because it seems to have crashed. So just give me a moment. Okay. Right, let me try and share that again. Okay, it's working again. Magic. Okay, so yes, we're open to a lot of collaboration. Like Steve said, we've set up many workshops over the past couple of years to try and foster collaboration, match people up and get some funding opportunities off the ground. One example of an opportunity is our Centre for Doctoral Training in Advanced Biomedical Materials. So this is funded by the EPSRC, and this is to recruit around 54 students over 5 years to do 4-year programme of study for their PhD. So if you have any ideas, one good way of linking up and starting a project is to put in a project proposal for the CDT. So the timings are, we usually ask for project proposals at the end of October. We do an internal sifting of projects, and then the successful projects are advertised in December, and we have supervisory team interviews. We then nominate the best candidates for the projects, and then they go forward to a CDT panel interview, which is usually in March. This is a programme that's joint with the University of Sheffield, and we've got many companies on board that collaborate and act as industrial partners. And we've got a lot of overseas institutions where students can be hosted as student placements. So the students get an opportunity to do an approximate 3-month placement in a company or an overseas institution or anywhere really that's going to enhance their employability. So we very much like to have co-creation of projects between academics and clinicians, and where possible, industry as well. So many of our CDT projects are have clinicians as co-supervisors. And if we have industry partners on board, then they provide either 5K or 10K a year, depending on whether they're an SME or not. And then we get them to participate in our many careers events and training opportunities for the students, and maybe also host some student placements. So do get in touch if you're interested in putting forward a project for the CDT. Like Steve said, there's a lot of funding opportunities within Manchester, and a lot of these come through the Translation Manchester team, and a lot of them are on their website. So we have some accelerator awards. So these include access to expertise and confidence in translation. We've got some University of Manchester schemes where we have gained funding from some of the charities and research councils. So for example, we've got the Wellcome Trust ISSF, we've got MRC funding, BBSLC funding, and EPSLC funding for impact accelerators. And of course, we've got the usual funders, including NIHR, Innovate, etc. So we do have advice and support available for people to help them identify appropriate schemes for funding. So please do get in touch. So like Steve said, you're welcome to contact us. We've got a website and of course an email address, amm@manchester.ac.uk. And Nicola will put you in touch with whoever you need to be put in touch with. And we're on Twitter, and we're on Pure as well. So please do get in touch if you have any questions or collaboration ideas. And I think I'll leave it there, unless Steve has got anything else you want to say.
No, nothing to add up there. Just open the floor to questions if anyone wants to ask anything.
Okay, I've stopped sharing my screen. So yeah, any questions, anybody?
Can I ask a question? Peter Clayton here. Really interesting and informative presentation. So thank you very much. So I'm going to ask a slightly tricky question. Where's Manchester placed internationally in this sort of area? Are we right there at the top? Are our facilities better than anywhere else? I know you mentioned, Stephen, that some of the facilities we have are really cutting edge. So is this an area where we are internationally recognised?
Yeah, absolutely. Yeah, a lot of our researchers are definitely internationally recognised and considered as leaders in the field. We collaborate widely internationally. We've got a lot of collaborations, for example, especially in Melbourne, Toronto, in India and the States. and various places in the EU. So yeah, I'd say Manchester is definitely one of the places to go to for biomaterials or biomedical materials research. Also recently, interestingly, I meant to say earlier, we've just got recognised as a facility for the European Space Agency as one of their 3D printing or bioprinting facilities. So that's quite exciting. Hopefully we'll be able to gain some funding to do some tissue engineering in space research and such like. So that's quite an exciting new development, thanks to Marco Domingos that Steve mentioned earlier. So
yeah, I forgot to put that in as well. I think it's only something like 5 places across Europe, isn't it, that they're partnering with. So the Manchester is the only one in the UK.
Yeah, so that's a good example where we stand in the field as well. Yeah.
And I think, Peter, just if I follow up on that, not from an international perspective, but from a national perspective, if you look at the metrics around publications and impact, Manchester, the trajectory over the last sort of 10, 15 years in this area, we've accelerated a lot. We're right up there now in the UK as one of the leaders alongside places like Imperial. So, you know, if you look at our trajectory, it's definitely on an upward slope and continuing to go up as well. And we definitely recognise internationally, as Julie says.
Great, thank you. Thank you. And is there more that we can be doing for you for clinical links? You may recall that MIMIT was set up about 10 years ago to identify unmet clinical needs and then link them up to researchers and companies to get those unmet clinical needs addressed. Now that was wider than what you're doing, but and there was a sort of Jack Eldham originally set up a network of site miners in the hospitals to kind of go round and talk to clinicians and identify ideas. And I don't know, is there more that we could be doing to highlight what you're up to and getting more ideas coming through? Because you seem to have at the moment a really good track record in the translation through to a spin out, which is fantastic.
Yeah, definitely. I think there's always more that can be done. I mean, we try hard to get links with clinicians. We've got a few clinicians that we rely on quite heavily again and again. So we're always welcome to finding new clinicians and new links. Like we said, we do run these workshops that we try and advertise as widely as possible. So the cardiovascular one and a co-cultures and organoids one, a cancer one, and a graphene one coming up. So we try and get clinicians to come along to those, because that's really where we try and join people up and talk about new projects that we could perhaps move toward funding applications. So ideas on how to reach out to more clinicians would be very welcome, or ways to advertise our workshops, or even, you know, vice versa. You know, absolutely like clinicians to come to us and say, look, we've got this problem. There's no ideal solution at the moment. Can you do anything with the tissue engineering or whatever approach? And we'll just, because of the breadth of work going on in Manchester, we're pretty much covering all tissues and organs, I'd say, Steve, wouldn't you?
Yeah, and I think, just follow on from what Julie said again, you know, that we have a huge network across Manchester and probably no shortage of ideas of potential solutions. I often think, you know, every idea is a good idea. You know, if there's a need there, we can try and at least find a solution to it. And I worry that sometimes the university can seem quite a daunting place that is so big and so sprawling. What we'd like to try and think that we can do is be that gateway in. So it's how, as you asked me, how do we reach out to clinicians who aren't necessarily interested in the research side of it directly, but have a question where we can try and help them come up with a solution. So we have some booklets that we can pass on as well to try and disseminate, just with, you know, ideas in the bioelectronics on the biomaterials angle and different projects that we've got going on, just to, a bit like we tried to do today, stimulate ideas and thoughts of, oh, that might be quite useful. And then we can follow up with more conversations and more detail. So we can certainly share those around. it's in the absence of Jackie and Mimit, and because we did talk to Jackie at one point, and then Mimit, I don't know what is no longer, or I don't know why.
We've folded Mimit into Health Innovation Manchester.
Yeah.
So the potential is still there, and the site, there are still site miners around.
Yeah, and we've spoken to Jonathan Massey as well, and to others, and I think that's part of what we want to do now, that there's a bit more happening on campus and around again, that is try and really kind of push forward with a lot of those ideas of reaching out more and linking in more with clinicians.
Yeah, many of the hospitals have grand rounds, and that might be an opportunity to just go and do a sort of presentation that you've done today, actually in the hospital settings.
I'm certainly happy to do that as well. If there's opportunities we can.
Well, maybe we can have a talk about how we could link you in to that.
Yeah, that'd be great. Yeah. Any other questions from anyone? I don't think there's anything in the chat, but just a comment from Jonathan, which I think the inclusion in the BRC I think is really important. If assuming it gets funded, hopefully it will, and it will allow us again to kind of get our name better known in that community as well.
Yeah.
Yeah, thanks to Sinead for mentioning the MCRC. Yeah, we are trying to broaden our links with various people at the Christie. So, but obviously the more the merrier. But yeah, we'll get in touch with you, Sinead, for sure.
Yeah, and AMM BRC PhDs. Yeah, I think PhD projects are a great way of. Sorry, that was a comment from Jane. A great way of kickstarting work that leads to bigger funding pots. And there's obviously the CDT is running at the minute, but it's time limited. So there's only, there's a limit to how many students we get through that. So yeah, anything moving forward in terms of studentships would be great. Okay. Last chance for any questions, I guess.
Yeah, so I don't think there are any other questions. I think we've gone through the chat. I think so. Thank you both for giving the presentations today. I think that's been really useful, and look forward to working with you both more closely in the future. So thank you everybody as well for joining, and hope to see you in the next MAHSC seminar.
Yeah. Great, thank you everyone, and we'll share the slides around. I've already contacted Jane with the slides, so we'll get those shared around.
Thank you.
Thanks very much.
Thanks very much.
Cheers now. Bye bye.